Welcome everyone to this live webinar on advancing vaginal health products. Today we'll explore how next generation multipurpose prevention technologies or MPTs are helping address bacterial vaginosis, sexually transmitted infections, and HIV prevention. We are honored to be amongst distinguished presenters and individuals today. To Doctors Jen Balkus, Ishita, Shah, and Laurel, Lagenaur, Dr. Bethany, Holt, and of course you, all the participants joining us from across the globe. Thank you for joining us and for contributing to this important conversation. Now, without further delay, allow me to get this session started. This live webinar is hosted by the Initiative for Multipurpose Prevention Technologies, or IMPT, and we encourage those that want to connect to please introduce yourselves in the chat. You can share your name, location, email, whichever is comfortable. I also want to remind all of us of a few housekeeping rules. So all participants will be muted during the presentations. However, if you do have any questions during the presentations, please use the chat box and we'll address them in the Q&A session. When we reach the discussion, you're welcome to raise your real hand or your Zoom hand, unmute and participate. We do encourage open dialogue and we look forward to your engagement. Let's have a brief look at the agenda for today. So my name is Claudia van Wyk, and I'll be guiding you through the introductory portion of today's webinar. We'll hear from these three experts in the field over the next 45 minutes, where they will each share insights from their work at the forefront of vaginal health, antimicrobial therapeutics, and HIV STI prevention. After the presentations, Dr. Bethany Holt from the IMPT and CAMI Health will moderate the discussion, providing an opportunity to explore key themes and emerging innovations. We'll open the floor for Q&A, and as I mentioned earlier, we encourage you to ask your questions and share your perspectives. We always look forward to a meaningful and engaging session with all of you. So the Initiative for Multipurpose Prevention Technologies was launched in 2009 and serves as a global product neutral platform dedicated to advancing MPT development. IMPT brings together over 2,000 stakeholders from across nearly 24 countries, creating a collaborative environment that provides technical guidance, strategic direction, and scientific leadership. By connecting researchers, product developers, funders, and advocates, IMPT plays a critical role in accelerating innovations that address HIV prevention and broader sexual and reproductive health needs. In 2020, IMPT launched the Let's Talk MPTs webinar series to foster ongoing dialogue, share research advancements, and strengthen collaboration across the field. Today's session is part of that continued effort to advance innovation and improve global health outcomes. The MPT Resource Center is designed to support the field across the entire MPT ecosystem, and at the core is our MPT product database. It's a searchable product pipeline tracking candidates across indications and stages of development, and we are currently conducting our annual update to ensure the data remains current and comprehensive. We also house several focused knowledge hubs, including the Advocacy Hub, MPT Regulatory Guidance Hub, as well as the Socio-Behavioral Research Hub. Beyond these technical resources, we also feature digital stories, which elevate lived experiences and community perspectives within the MPT landscape. For professional growth, we list MPT trainee and career resources, as well as regularly updated funding opportunities to support research and development efforts. The link is on the screen, So we do encourage you to go have a look. In partnership with key stakeholders, we outline MPT field-wide action areas, which identify strategic priorities for coordinate progress across the sector. And finally, we keep the community connected through our conferences and events, MPT learning events, and webinars, which are available on demand. And of course, the bi-monthly newsletter, the MPT Insider, which provides ongoing updates and highlights. Today, we are absolutely honored to learn from 3 leaders whose work spans epidemiology, microbiology, and innovative product development. We have Dr. Jen Balkus. She is an infectious disease epidemiologist specializing in sexual and reproductive health and health equity. She serves as a senior epidemiologist for the HIV, STI, HCV program at Public Health Seattle and King County, where her work focuses on understanding communities most affected by HIV, STIs, hepatitis C, and developing innovative prevention and communication strategies. She's also clinical associate professor in the Department of Epidemiology at the University of Washington School of Public Health. She's led NIH-funded clinical and observational studies on vaginal health and STIs in the US and globally, alongside teaching and equity leadership in public health. We have Dr. Ishita Shah. She's the CEO and co-founder of Matrubials, a biotech company developing milk-derived antimicrobial therapeutics to combat antibiotic resistance. She has extensive experience in bacterial pathogenesis, antimicrobial discovery, and the biology of human milk components. Before founding Matrubials, Dr. Shah served as Associate Director at the Foods for Health Institute at UC Davis. She collaborated on research into bioactive milk peptides and human milk oligosaccharides. She earned her PhD in microbiology from the University of Maryland, Baltimore County, where she discovered a novel mechanism of bacterial transcription activation and received the Nat Sternberg Thesis Prize for outstanding doctoral research. Next, we have Dr. Laurel Lagenaur, recently appointed as Osel's Vice President, Women's Health. I would also like to add she was Osel's first employee and has led the development of Osel's lactobacillus technology platform as the Director of Research. She was the principal investigator on numerous research grants, including Osel's first SPIR grant to develop Mucocept HIV as a novel live biotherapeutic to prevent HIV transmission in women. Dr. Lagenaur is a molecular biologist with extensive experience in virology and bacteriology. She received a doctorate in microbiology and immunology from Stanford University and completed a postdoctoral fellowship at the University of California, San Francisco, before joining Osel. Together, their work represents critical advancements in science, clinical research, and public health. I'm thrilled to hear from them and beyond inspired by these individuals. So, Jen, I'm going to unshare. The stage is yours.
Great. Thank you so much, Claudia, for that wonderful introduction and to set the stage for today. And I'm so excited to be here with my co-panelists. Let me just hit this last button. And I'm here today really wearing sort of my University of Washington hat, talking about one of my favorite topics, which is bacterial vaginosis. And it's such a pleasure to talk about this condition in the context of HIV and really think about broadening the conversation, to think about this relationship and what do the role of multi-purpose technologies could they play in terms of both reducing bacterial vaginosis and potentially reducing HIV and STIs as well. Before I keep going, I just want to make sure the slides are viewing okay. Great, thanks, Claudia. So briefly, what I want to do is provide an overview that will help us with our conversations and help support the presentations that my co-panelists will be giving as well. So we're going to focus on the epidemiology of bacterial vaginosis, otherwise known as BV, talk a little bit about methods to characterize BV and the microbiota. I'll touch on current BV treatment recommendations and challenges with those given the high levels of recurrence, and then just briefly touch on the relationship between the microbiota and STI acquisition and some of the methodologic challenges that emerge when we're trying to look at these two relationships that would impact our ability to evaluate the impacts of MPTs. And before I get started with this, I just want to give a little bit of context for the data that I'll be sharing today. So most of the data come from studies about the vaginal microbiota and BV among cisgender women or individuals presumed to be cisgender women if gender data wasn't robustly collected. There are data that exist on the penile microbiota in cis men, particularly with its relationship to BV recurrence and the impact of partner treatment. I'm not going to have time to cover those today, but do want to call your attention to their existence in the literature. And I also want to highlight an important gap in the field when it comes to thinking about vaginal health in the microbiota, and particularly related to both transgender and cisgender, or transgender in women and transgender men. There are studies related to the microbiology of the neovagina among trans women, as in a growing literature on the vaginal microbiota among trans men. But an important gap is that there is no current research on the impact of whether changes in the microbiota in these two important populations impact STI and HIV susceptibility. It's an important area of further research that we won't get to touch on today. So globally, BV is the most common vaginal infection. It affects hundreds of millions of women each year across the globe. It's a condition that's characterized by watery or white-gray discharge and a fishy odor. And these symptoms really represent a shift from a lactobacillus dominant environment to one that's dominated by anaerobic and gram-negative or gram-positive bacteria. There's a number of studies that have looked at the prevalence in different populations, and we see prevalence ranges from anywhere from about 20% to greater than 40% among reproductive age cis women, depending on the population under study. This figure here comes from a review by Catherine Peebles et al. That looked at the prevalence of BV, kind of trying to get at the general population estimate rather than specific targeted populations. And we see that about a quarter or up to a third of women in each region have a prevalent BV at any given time, which is an incredible, incredible burden of BV across the globe. And since this is a clinical condition, it's really helpful to understand sort of a little bit more microbiologically what's happening that results in these symptoms and what is sort of our healthy or optimal environment versus one that is indicative of potentially of BV. So the next few slides have figures from a recent review by Catherine Bradshaw and colleagues that really help to elucidate these findings in a really clear way. So we talk about sort of healthy or optimal here on the slide, where you can see that there's just really one or very few types of bacteria, and it's the lactobacillus dominated bacteria, which are these sort of purple rods here. We see a low pH, so usually less than 4.5. There's some metabolites, there's peptides, there's some anti-inflammatory cytokines, but there's very few other sort of immune cells. And we also see a nice, usually thick mucosal barrier that manifests either as like a clear discharge or sort of a normal, quote unquote, normal discharge for the individual. And that is all sort of there to help maintain this really solid and healthy vaginal epithelium. In the context of sort of a less optimal or suboptimal, or sometimes the diagnosis of BV environment, we see something very different. You can see that this image is much busier than the last one. There's a number of different bacteria that are present. The pH increases. There's various amines and metabolites that are present. We see pro-inflammatory cytokines and other immune mediators being produced, and the recruitment of immune cells as well into the epithelium. We lose that sort of mucosal barrier, which can then help disrupt the epithelium, creating potentially breaks in the epithelium. So all of this is sort of happening on the microbiologic level that then manifests into this gray, white, watery discharge or amine odor that's reported by women. But it's important to note that in this context, only about half of women will report symptoms of BV, even though these immune microbiologic and immune changes are happening under the surface. So as a clinical condition, it's diagnosed in a number, several ways that have evolved over time. The first method that's been used to diagnose BV is Amsel's criteria. So it creates consists of 4 criteria: pH, vaginal discharge, a whiff test, which looks at using potassium hydroxide, and then clue cells, which are here on the slide. And a clue cell is really that epithelial cell in the right box that is sort of coated and covered with bacteria. So that's why it looks so fuzzy there on the slide. The Nugent score emerged as another way. This is typically used in research settings and not in clinical settings. And it involves a gram stain to look at the morphotypes of bacteria present, and there sort of ends up being grouped into 3 categories after it's scored. And you can see normal there. We have epithelial cell in the middle with these, again, longer rods, which are indicative of lactobacillus morphotypes. Intermediate, where we see fewer of those long rods and more of the sort of short rods there. And then BV, where we just see in this particular image epithelial cells that are coated in abundance of anaerobic bacteria. There are several rapid point of care diagnostics that are highlighted here, such as BV Blue and the FEM exam card, which use components of the AMSLS criteria for diagnosis. And more recently, we've seen the emergence of PCR-based assays, which use algorithms based on the bacteria detected to make a determination of BV versus not BV. And I think one of the key things that's emerged over the past 2 decades in the field of sort of vaginal health and BV is, I think, both a confirmation and an expansion of our understanding of the heterogeneity of BV. We as the scientific community has been studying BV for over a century, and really early methods using culture. And challenges with culture, often involve that you have to be able to grow the bacteria to see it. And with PCR-based methods, we can culture much more robustly, especially those bacteria that are very different or to grow. We can assess those. And you can see here on the slide, these are both images from Sujatha Srinivasan's group and Jacques Rebel's group, looking at just the composition of the vaginal microbiota. And it's quite diverse in general and between individuals. So no BV sometimes is the same. You're looking at different microbiologic compositions that still might manifest in the same symptomology. And it's also highly variable over time, which creates its own other challenge. And we see these up and down shifts that result in different symptoms and in response to treatment, and as well in response to different behaviors that folks might be engaging in. So there's a lot of challenge in terms of understanding these relationships when we start to think about mechanistic relationships between bacteria that are present and other sequelae. So as a clinical condition, BV there's treatment for BV. There's here the sort of the high level summary from this US CDC, European Union, and WHO. All 3 bodies call for treatment of symptomatic individuals that are symptomatic, and I'll talk about the exact treatment methods in just a second. And the one sort of difference here is that in the EU, treatment is recommended for asymptomatic individuals who are pregnant with a prior history of idiopathic preterm birth or second trimester loss. And the goal of treatment here, again, is clinical. It's to alleviate symptoms and not necessarily look at any sort of changes microbiologically. So as noted, we're really focusing on treating individuals who are symptomatic, and with the sort of the top 3 treatments that are recommended, most commonly metronidazole, oral metronidazole. There's also metronidazole gel and clindamycin cream that can be used as well. We see cure rates of about 75%, which is good, but not great. That's a lot of individuals who still experience symptoms after a course of treatment. And most importantly, it is the level of recurrence that we see. We see about 30% of individuals will have a recurrence. So again, having symptoms come back within about 3 months. And in a study by Kat Bradshaw's group in Australia, looking at this more closely, they saw 23% of individuals who were treated for BV saw their symptoms go away, experience recurrence within 1 month, and nearly 60% saw a recurrence within 12 months. So that's a lot of BV that's coming back repeatedly. And there's a number of factors that potentially impact recurrence. And just I think it's important to touch on these a little bit here, because again, this sort of gets into thinking about mechanisms for disruption. So we try to separate these concepts of recurrence and persistence out separately, because they're the sort of the strategy or the interventions may differ for trying to deal with both. So recurrence could be potentially you clear the bacteria, you see the symptoms go away, but you may not recolonize with those optimal lactobacilli bacteria, which kind of creating a gap. So if there's this re-exposure to potentially, bacteria from a sexual partner, then you would see that potentially recolonization with BB-associated bacteria rather than lactobacilli. There's host and genetic factors that play that impact both recurrence and persistence. I think one of the big challenges with thinking about persistence is that full eradication of bacteria. So we're always thinking with any antibiotic exposure about antimicrobial resistance. There's evidence that certain bacteria that are associated with BV may form a biofilm, so really kind of become adherent. So they might decrease to low levels within the epithelium, but not enough for eradication. And as soon as that sort of pressure from the antibiotics is gone, may reemerge, and other factors that potentially impact persistence as well, such as the copper IED and others. So it's a really sort of complex pathway that drives this repeat BV positivity following what appears to be successful treatment with the eradication of symptoms. And so BV is, as I think a condition that has been very underappreciated in the field of women's health, because it manifests, I think, in this way of quote unquote just discharge. There's sort of this dismissal of the importance of the role of it, both in women's health in general, and its larger role with relationship with other sequelae. And so I think it's important to focus on the importance of it on its own in itself. And there's some great qualitative work related to this, where women who especially have recurrent BV talk about experiencing shame and stigma, incredible frustration and helplessness for sort of having this reemerge over and over again, and a lack of control over recurrent BV, and then having a direct impact on sexual behavior. This is a quote from one individual in the study talking about how it makes her feel more insecure. Even though you're not having sex, you don't want to be close to people because they might smell you. BV-associated, importantly, have BV and BV-associated microbiota have also been associated with increased multiple serious health outcomes. And so we see a number of studies that have looked at this related to infertility, adverse birth outcomes like preterm birth, STIs and HIV, HPV, as well as gynecologic infections and surgery. And so one of the things I think that has been a huge challenge in trying to move the field forward with these relationships of BV and specific microbiota and STIs, including HIV, is both determining this causal relationship and evaluating this in terms of, is an intervention going to successfully address this? So I'll walk you through this sort of the concept. I think it's important to think about this for next steps. So if we have individuals who are potentially at risk for an STI, for example, and we randomize to an intervention, we'll have our folks who get the intervention, folks who get the control. And if this is an intervention that's trying to address either BV or microbiota, the first thing we need to do is modify that environment. We need to modify that environment enough that in step one, that we would actually see if there is a true causal relationship, a change in step two. One of the huge challenges, as we've noted, is this issue with the interventions that we have already for BB. Is if we have recurrence rates of 30% by 3 months, if we are only successfully treating 75% of people, it's highly possible that we could have a great rigorous study and see no change in STIs, and have it not necessarily because there's no causal relationship, but because we haven't adequately or durably changed the vaginal microenvironment in a way that it actually then impacts that level of susceptibility. So being able to look at this sort of in a phased approach and know that we have an intervention that can appropriately and significantly, in whatever way it needs to, modify that environment to then have an impact on the STI or HIV or other sequelae is a critical important to be thinking about. How do we evaluate either individual interventions or multi-purpose interventions to successfully achieve the goals that we're trying to achieve? We tried to address this in a study that we conducted a few years ago now with Scott McClelland about monthly periodic presumptive treatment for vaginal infections. So here it was trying to get at the issue of recurrence and durable changes, where we randomize individuals to either a placebo or monthly periodic presumptive treatment for BV and miconazole for vulvovaginal candidiasis. So individuals took the treatment regardless of whether they had BV or not. And we saw a 35% reduction in BV over the course of the 12 months. And we leveraged the samples that we had stored and collected as part of the study to look at STIs, and the impact of the intervention on STIs. And we're not powered to look at this as an exploratory analysis, but I think notably what we observed is that we saw, at least at the point estimate, decreases of chlamydia, gonorrhea, and mycoplasma genitalium among individuals randomized to the intervention arm versus the placebo. None of these were significant on their own. They were when you combine them, but that's just kind of due to study power issues. But I think it's an intriguing and notable observation enough to say like this is potentially could be possible if we had the right intervention and sort of looked at this more broadly. So it was exciting and I think an important area to consider in the field going forward. So with that context, I think it's important just to think about these sort of bigger questions that can MPTs with the sort of the right mechanistic relationship disrupt these relationships between the microbiota and STI susceptibility. So we used a blunt tool, which was just high dose metronidazole, and we observed this decreased incidence of several STIs. Could targeted intervention or something that sort of operated with multiple mechanistic pathways have a larger effect on susceptibility? And what is it exactly do we need to target? Do we need to be thinking about specific bacteria for both reduction or promotion? Do we need to be thinking about an entire community shift, or are there other sort of pathways such as disruption of metabolites or pro-inflammatory cytokines that could be an approach to look at this? And noting that, on the slide, there's a number of pathogens presented, but the mechanistic relationships between the microbiota and each of these pathogens may not actually be the same. So it might be a different strategy if you're trying to reduce, gonorrhea versus trying to reduce HIV, or potentially something that could be more broad and could operate in the same way. So with that context, and given the global burden of BV and STIs, both across really in every region, including the United States, that we see amongst cis women and other individuals assigned female sex at birth, a safe and effective MPT could have a significant impact on sexual health. And I'm delighted to pass things off to my colleagues to hear more about the work that they've been doing to try to really address this issue. I will stop sharing here. Okay, Ishita, over to you.
All right, wonderful. Can you see my screen? Yeah, okay, well, great. Thanks, Jen, for such a wonderful introduction. And I'm really honored to be in the room with Jen and Laurel and all of you. I'm going to be talking today about our work on specific development of antimicrobial peptides that are milk-inspired for the treatment and/or prevention of bacterial vaginosis and other STIs. And this work has been done at Matrubials, which is a spin-out from the UC Davis Foods for Health Institute and Milk Bioactives program, where my co-founders, Professors Bruce German, David Mills, and Carlito Labriglia, have dedicated more than about 20 years studying the structure-function relationships about human milk and other mammalian milks and the impact on infant and maternal health. And together, we have worked on the specific activities of bioactive milk peptides and oligosaccharides prior to founding the company based on the observations we made. In terms of our core mission, we are addressing antimicrobial resistance, which is a common healthcare threat, where even in the pre-pandemic year of 2019, there were 1.2 million deaths that were attributed to drug resistance. And the antibiotic development pipeline continues to remain weak, which is why we need to come up with more safe and innovative therapeutics that result in similar activities as antibiotics, but also lesser side effects. And in terms of human milk, as we're all aware, it's sculpted by evolution to not just be nutritive, but also protective for the newborn. And that's true for all mammals. We had some early indications based on flash fractionation chromatography of human milk that resulted in pools of natural peptides that are present in this milk. And what I'm showcasing here to you is that when we subjected enterohemorrhagic E. coli or clinical isolates of Pseudomonas to some of these pools of peptide fractions, we were able to demonstrate that there is effective bactericidal activity based on the activity of these peptides in those pools. And what we observed is that based on sequencing outcomes, that each of these pools had about hundreds to, in some cases, a total of a few thousand peptides that in terms of a scalable technology or batch-to-batch variation from flash fractionation is not possible to translate for clinical development or safety aspects. But we learned that there were several short peptides in these pools and that there is a way of synthetically manufacturing them to be able to develop them clinically as lead active pharmaceutical ingredients. That led us into sort of getting inspired by human milk further to develop an AI-enabled platform that would accelerate our ability to discover numerous milk antimicrobial peptides that can be diverted into the clinical pathway for development and validation. The way that process works is as input, we use dominant mammalian milk protein sequences. So these are beyond just human and bovine, that could also include camel, sheep, cow. And we found out through the specific code that we input is that we got a data set of thousands of peptides that are a particular length that we would want as part of a translation towards a business model with specific cost of goods. These are all natural amino acids that can be made into large-scale synthetic peptides, and that they were asked to be of a specific charge, mostly cationic in range, and would have a specific secondary structure in solution so that we can make predictions about their activity and safety in vitro and further in vivo. This library of thousands of short peptides was then navigated through selecting candidates that were passed on for preclinical activity testing, as well as de-risking from points of view of minimal inhibitory or bactericidal concentrations or time kill assays, both with pathogenic bacteria as well as commensal bacteria. And in vivo safety tests that were hemolytic assays using red blood cells or host cell viability, as well as human organoids, depending on which specific indication we would be interested in. And so the first FDA-regulated product that we are building here is a vaginal insert to address bacterial vaginosis and additional complications that also include HPV, and I'll touch base upon some initial work we have with that. And here is an example of a 3-gram vaginal insert that can be self-administered, but can be prescription-based and reimbursed by insurance. The typical US TAM ranges between 4.8 to 5 billion, based on at least 21 million women in the US being diagnosed with bacterial vaginosis on a yearly basis. Jen did an excellent job about talking in a great detail about BV, so I'm not going to go through this slide again, but it is true that the impact is really high with BV as the starting point and the changes in the pH and epithelial integrity, predisposing now these women to secondary complications that are not limited to infections, but also reproductive challenges. Together, this umbrella impacts nearly 1 billion women globally with just 2 antibiotics that are still made available to patients. And one of the reasons I want to talk about this slide is because on an average in the United States, even in this day and age, there's obviously at least 1 in 10 babies that can be born preterm. With BV, the increase in preterm birth risk is about 2 to 3-fold higher. And in that case, what happens is that these babies are highly vulnerable and they're already predisposed to additional complications that are neurodevelopmental in order, but also early allergic issues, as well as one of the most debilitating diseases in the NICU hospitalized cases, which is relevant to necrotizing enterocolitis and less representation of commensal gut bacteria for these patients. So there is a need to address a lot of these complications, and coming from the program at UC Davis, where maternal health has been of primary importance, it's dependent on good reproductive outcomes as well. And so we're really very interested in continuing to work in that aspect of addressing just not just BV, but also the reproductive challenges that are associated with it. And again, I will quickly go through just this slide describing metronidazole and clindamycin. They're both available for oral use as well as in terms of direct vaginal use, with metronidazole being the most commonly used drug and clindamycin available only in parts of the world, but not to every patient who is in need. With clindamycin, unlike metronidazole, it also impacts the healthy vaginal microbes as well as healthy gut microbiomes, resulting in a lot of these patients having gut-related side effects, which they suffer from for a few weeks to a few months and end up not having to take that again and again, even if there is a strong need. So in terms of antimicrobial peptides, one of the key differentiation factors that we've seen, and again, similar to metronidazole, is that they're selective for the pathogenic bacteria associated with BV. So we talk of Gardnerella vaginalis, but also Atopobium and Prevotella and Megibacillus indollicus and so on and so forth. But as is expected for peptides, there is low or no resistance in the assay systems that we use, which is a huge benefit as compared to standard of care antibiotics, where resistance can start to happen within a few years of introduction of a new antibiotic. I'll also discuss quickly the safety aspects of some of these peptides that we've tested, and most importantly, that they're synthetically manufactured through solid-state chemistry with the advancement in the technology, where we're able to obtain gram quantities within just a few weeks that can be scaled for lead API clinical development. These molecules are also IP protected for composition of matter, but also efficacy and safety data. What I want to also talk about is that in addition to our in vitro work, we were able to evaluate vaginal swabs from patients who were diagnosed with bacterial vaginosis at an OBGYN clinic, AIWHC, locally here in Sacramento. And one of these examples is shown here where, compared to the no treatment versus our 4 candidates being tested in these vaginal swabs, we observed over and over that in many cases, patients had samples that were fused with pathogens that were resistant to both standard of care antibiotics, metronidazole and clindamycin, and resulted in a certain viable count, whereas there was dramatic reduction in presence of all of our 4 candidates. Now, this work is important because not only is it about ex vivo efficacy in the absence of a very robust BV animal model, but it also told us that in diverse microbiome, that is because of different patient samples, but also different mucus compositions, our peptides were still able to showcase the efficacy. And in addition to that, our collaborators at Conrad evaluated some of these peptide candidates for their effects on HIV-1 and HSV-2 and demonstrated exciting antiviral activities as well. So we're kind of thinking of this as not just to impact BV, but additional secondary complications that may be associated with BV. And in work that I don't have time to talk about today, we showcased in this ex vivo study that several patients also had comorbidities with Candida albicans, where neither metronidazole or clindamycin were impacting Candida, but all 4 of these peptides were able to completely result in loss of viability of Candida as well. Following that, we built a 0.5 gram prototype, which was a vaginal suppository specifically designed for a 10-day rabbit vaginal irritation test, where daily symptom check and end of study histopathology were useful in determining that unlike an irritant, which resulted in vascular congestion or epithelial thickening or infiltration of leukocytes, our peptides at concentrations of 5 and 20%, which is the highest solubility, were deemed to be safe. There's no resulting irritation in these rabbits. And based on the in vivo safety as well as the in vitro MIC-MBC assays, that tells us that there is an attractive therapeutic index and the capacity to translate these peptides for the purposes of reducing the polymicrobial biofilms associated with BV. Now, if you remember, Jen described earlier that in patients with BV, what we do know is that there's a dramatic reduction also in the antimicrobial peptides, which serve to protect the vaginal epithelium. So in a way, this can act as restoration of antimicrobial peptides by on-site high-dose delivery so that we can disrupt those biofilms that are so notorious in terms of recurrent infections as well. So in summary, based on sort of an accelerated discovery platform from human milk, we now have 2 specific candidates that are developed further for IND applications, where we not only see safety from epivag organoids in rabbit studies, but also reduction in host inflammatory outcomes during an active infection of the epivag organoids. And we've been able to submit a briefing package to the for a pre-IND application to the FDA, as a result of which we now have guidance from the agency relevant to the final stages of IND-enabling studies, which include GMP manufacturing, a full CMC package, and GLP tox of these peptides as a way of developing them clinically towards a new drug approval, so that these vaginal inserts can be commercialized for a first-in-class way of delivering antimicrobial peptides, which can then be a prescription model and reimbursed by insurance. And that would be our early way of getting to the market to start with at this time. So in terms of pathway through FDA to market, we like to think that we are halfway there, with the preclinical early R&D and de-risking being done and getting to the IND-enabling stages, where work has been funded by SBIR phase 1 grant from the NIH, the ex vivo studies by the Gates Foundation, and Y Combinator and others for private funding that can now lead to IND application once we are able to wrap up the final stages of that. So we're very excited to continue on this path, where we can then envision clinical development with a CRO and or a strategic partner towards launch of a new product. And in terms of the business aspects of it too, we do believe that the revenue projections that we have, based on assumptions of percentage penetration starting at 3 to up to about 10 within 10 years, can result in ways of really good return on investment for groups that can fund these kinds of projects, because antimicrobial discoveries or infectious diseases or women's health do need to have a lot more focus on further funding so that we can come up with safe therapies for women. And I'm happy to discuss any of these in detail with anybody who's interested in terms of how we come up with these numbers. So just the last one slide is about our current portfolio at Matrubials, is where women's health is our lead indication, and we're excited with 2 candidates, one that's specific to BV and the other one is BV, but also shows neutralization of HPV 16 and 18, which are commonly associated with cervical cancer. So we are further doing mechanistic studies through grant proposals for the second candidate. But we also have a lead candidate for skin and soft tissue infections that we have in vivo efficacy data, specifically from dermal infection studies with MRSA, that we are continuing to develop as well towards IND applications. And we have a consumer product that we are evaluating the launch of that has another group of milk peptides, but also secondary ingredients that are inclusive of milk oligosaccharides and milk fatty acids as part of human skin regeneration aspects. And a really good group of advisors and supporters, and specifically Dr. Dina Canavero, who's an OBGYN, and worked with us for the ex vivo analysis and patient sample collection. And with that, I will stop. Thank you. I'm looking forward to Laurel's presentation.
All right. I don't know why this went to the second slide. All right. Today I'm going to talk about live biotherapeutics, and I'd like to give a couple of definitions. Live biotherapeutics are bacteria that are designed to treat, cure, or mitigate a disease of humans. And so it's the same definition as a drug or a biologic. The same is true for recombinant LBP, which I'll be talking about today. These are evaluated by the FDA via an investigational new drug program. They're marketed as drugs and prescribed by physicians. Probiotics are over the counter. They may or may not contain live bacteria, and they cannot make medical claims. You may hear the words like restores or maintains natural balance or supports immune function, but those actually are meaningless structure function claims. If you look at any probiotic at the bottom of the label, it will say these statements have not been evaluated by the FDA. This product is not intended to treat, diagnose, or cure diseases in humans. So we'll be talking today exclusively about LBPs, or live biotherapeutic products. I think Jen covered this well. I don't think I need to say much more about bacterial vaginosis, other than to remind everybody that BV does increase the risk of STIs and HIV acquisition, preterm labor and preterm birth, recurrent urinary tract infection, and HPV infection and progression to cervical dysplasia and cervical cancer. So Osel has been working to harness the vaginal microbiome to treat dysbiosis. Osel developed Lactin V, a Lactobacillus crispatus strain CTV05, as a live biotherapeutic product. Lactin V is a powder formulation in a pre-filled vaginal applicator containing about 10 to the 9th CFU of Lactobacillus crispatus. This shows you what the applicator looks like, and here's the bacteria. And the therapeutic indications for this are to prevent BV. We've been in phase 2 trials in the US and South Africa, and both of these have been completed and showed efficacy. We're currently in a trial for prevention of preterm birth at the Imperial College London in the UK, and prevention of HPV progression in a trial that should start soon with NCI. And finally, we've completed a small clinical trial for prevention of recurrent urinary tract infection. So Lactin V for prevention of BV, I want to give you a little bit of clinical history. In phase 1, it was safe and well tolerated at all doses. And in phase 2, it colonized up to 78% of women who used the product consistently. And colonization correlated with reduced BV organisms. In the phase 2B, sponsored by NIH, this was 228 women, prevention of BV at 12 and 24 weeks. We saw a statistically significant reduction in BV recurrence, and efficacy was correlated with colonization. In post-hoc analysis, women who achieved a clinical cure, meaning that their metronidazole treated the BV to begin with and cleared some of those BV organisms, we had up to 84% colonization in that group. And here are the papers associated. We also saw decreased inflammation. Here are the papers associated with this study, and the preliminary study phase 2 trial was published in the New England Journal of Medicine. Now here is a snapshot of the Lactin V trial. And what we can see, pre-metronidazole, most of the women, okay, let me start on this side. Women were treated topically with metronidazole for 5 days, and then they received Lactin V or placebo at a 2-to-1 ratio for 5 days, and then twice weekly for 10 weeks. And they were evaluated out to 24 weeks, which is 13 weeks post-dosing of L. crispatus. So here are the women pre-metronidazole. We see they all have what would be described as BV in general. I think one woman there doesn't. Post-metronidazole treatment, we see a large increase in lactobacillus eners, but very few women immediately convert back to a lactobacillus dominant microbiome. At week 4, when we start dosing, we start to see women pick up Lactobacillus crispatus, which is shown here in yellow. And if you look at week 24, again, this is 13 weeks post-dosing, you can see that women, what did this do? Sorry about that. Oh, it went the other way. Okay. Post-dosing, women in the placebo group very rarely pick up Lactobacillus dominans, but women up here that were treated with Lactobacillus crispatus, you can see a good proportion of them become Lactobacillus crispatus dominant. This trial is approved for phase 3 by the FDA, pending funding for when we've been looking for partners to fund this phase 3 trial. This work was recently published in Cell Host Microbe. Okay. And all right. So now I want to talk about engineering the microbiome to prevent HIV acquisition. So this concept we started a long time ago. Bethany knows the story. And it's called Mucocept. So it's modulating the vaginal microbiome to prevent HIV. So the concept is to colonize the microbiome with a lactobacillus to intercept HIV virus. So we genetically engineered a Lactobacillus jenseniae to produce a modified version of cyanovirin, which is a potent HIV entry inhibitor. We integrated this into the bacterial chromosome so that it's a single copy and it secretes cyanovirin. And it also has the properties of lactobacillus. It produces a D-lactic acid, lowers pH, is anti-inflammatory, inhibits BV-related organisms, and could potentially protect at the site where HIV infection occurs. So I want to talk for a minute about the inhibitor. We used an inhibitor called cyanovirin N that was identified at NCI in a drug screen. It's a very small, stable protein. It binds to specific high linkages of oligomannose on GP120. This just shows the structure, and this shows where it binds on the GP120 spike. It binds near where a CD4 binding site is, so it prevents the virus from attaching to cells. We purified this protein produced by lactobacillus, and we tested it against a CAVD panel of 63 tier 2 envelopes. And this included multiple clades and circulating recombinant viruses and transmitted founder viruses. And this cyanovirin produced by the lactobacillus inhibited 85% of these envelopes. And we have to remember that HIV, even though it's one virus with many envelopes. So you want to have the protein that you're expressing to be capable of inhibiting many different HIVs. And the average IC50 was about 2 micrograms per mil. So we wanted to move this product into the clinic, so we had to apply for an investigational new drug license, or an IND, for Mucocept. And it was formulated very similar to Lactin-V in an applicator to deliver about 10 to the 9th CFU of bacteria. And this was evaluated by both the CBER, the Center for Biologics and Research, and the Office of Therapeutic Products, because it's a recombinant product. So what does the FDA want to know about either your LBP or your recombinant LBP? They want microbial characterization, safety and resistance profiles, manufacturing and quality controls, and stability and storage of the product. So we did a lot of safety testing. First, we tested the bacteria and some formulated product. This was tested at a lab. This was tested at an independent lab that worked with human vaginal epithelial cells. They were inoculated. We inoculated either the parental strain or the recombinant lactobacillus expressing cyanovirin. We saw no induction of markers of mucosal inflammation, no induction of cytokines such as aromatase and MIP3 alpha, which would recruit T cells, and no up or down regulation of adhesion molecules such as ICAM1, which would be a marker of mucosal integrity. We saw no histologic changes in macaques that were colonized for long term with Lactobacillus jenseniae producing cyanovirin. And we did a 14-day rabbit vaginal irritation model. This showed minimal irritation that was similar to the placebo. And we also looked for antibodies to the recombinant protein, and we saw no IgM, IgG, or IgA in either serum or cervical vaginal lavage against the cyanovirin protein. And we also looked at activation of CD4 and CD8 cells as measured by CD69, which is an early activation marker. And this recombinant cyanovirin produced by lactobacilli did not activate CD4 or CD8 cells. So we wanted to manufacture the product for first-in-human testing. Again, it was manufactured just like Lactin V under GMP conditions. It met all the criteria that the FDA was looking for. It's delivered in a vaginal applicator, again, about 1 × 10⁹ CFU per dose. The FDA approved our IND because we demonstrated safety, biochemical identity, and genetic identity. So we showed that it secreted the modified cyanovirin, and the expression cassette was intact. And the stability of the product, very similar to Lactin V, is stable for over 3 years at 4 degrees, and in this case, -20. So Mucocept has moved, to a first-in-human safety trial, which is being conducted by UCSF. Here's the clinical trial number. Total of 12 women, 12 healthy women will be enrolled. In cohort 1, they'll receive 1 dose of Mucocept CVN. They're unblinded. We have 2 participants in this part of the study. We're assessing colonization and safety, looking for adverse events, endogenous vaginal microbiome changes and changes post-dosing, the pharmacokinetics, both tissue and systemic, and confirm clearance of the recombinant strain. So the first 2 women who were enrolled in the study following their single inoculation, go back. So first 2 women were enrolled. They've out of the way. They had their single dose, their samples were preserved for batch analysis, and they successfully cleared the organism, which is exactly what we wanted to see. In cohort 2, 4 women will be randomized 1-to-1 to receive either 1 dose of the Mucocept or the placebo. And again, we'll follow these criteria above. In cohort 2, 2 women will receive 3 doses of the Mucocept, which is unblinded, and will follow the same criteria. And finally, in cohort 4, 4 women will be randomized 1-to-1 to receive 3 doses of the Mucocept or placebo, and will follow them. So we're very excited about this clinical trial, and this is the first testing of a lactobacillus that produces a small protein and potentially could produce some of the peptides that were described in the previous talk, but to work against both bacterial vaginosis and HIV. And there are so many people involved in these studies. I've just presented some of the names here, and thank you very much.
Wow, thanks everyone. And Laurel and Jen and Ishita, thank you for your really wonderful presentations. I'm familiar with your work, and I always learn more when I hear you share. Yeah, so it's really great. I also want to thank all of you who have joined us, echoing Claudia and thanking you all for being here today. We are now going to open this up for question and answer, and you can enter your questions into the chat feature, as Claudia said, or part of really what we like to do with this webinar format is not just to learn from each other, but also to facilitate interactions and getting to know each other as a community. So feel free to raise your virtual hand if you'd like to ask your questions directly. Turn your faces on if you'd like. So as we'd love to hear from you. Claudia will put a link in the chat so that we can ensure that the MPT resources are as useful as possible for everyone. So as we're waiting to hear for some questions, I'm as moderator, I'm going to take the prerogative and ask one I'm really interested in hearing about from Ishita and Laurel. So the MPT regulatory issues and challenges are really, kind of the elephant in the room, a big challenge for the field. We're talking about products that address two or more indications, and the products in development also span, and include a variety of delivery types. So we've been for years sort of, working with our partners with, interacting with the FDA, EMA, other regulatory bodies. And I know that in your work, you have all had different, Shita and Laurel have had different experiences with regulatory bodies. So would be very I think it would be very useful to hear what your experiences have been. So maybe Laurel, you can start.
Okay, I guess when we started this, the FDA didn't have any guidance documents for live biotherapeutic products, but they did develop them in 2012, and there's been an update on that. The FDA has actually been very open, and more recently to regulating live biotherapeutic products. They're as usual, they're slow to respond when you are trying to get answers, but we've actually had very good interactions with them.
Oh, that's good to hear. Great. Ishita, would you like to
Yeah,
share?
I think in so many ways, same as Laurel, we typically based our briefing package for the pre-IND meeting with the FDA based on a guidance document that is typical for anti-infectives in any space, regardless of the indication that you're trying to serve. We tried to match with everything that is typically asked for in terms of efficacy and safety as part of requesting the agency's input on our proposal as to what we intended to do next as part of the IND-enabling studies, but also early clinical trials. And we got a response. It was the first round where we've been asked to essentially do what is typically referred as bioequivalence, but do our studies in a more GLP tox fashion versus a non-GLP lab, and evaluate that as part of the IND enabling pathway. And then, of course, the clinical trials are the ones where we want to make sure we have the appropriate design for the safety trial, but also an appropriate phase 2 trial where it's multi-site, multinational, and evaluates not just superiority or equivalence to standard of care antibiotics, but also are we able to really prolong time in remission with our intervention or not. And Bethany, as you have talked about before, the way we visualize our therapeutic would be that it's required to first sort of clear out the infection. But as was the topic of discussion in terms of really the impact on further outcome of disease and also STI, the restoration of the healthy microbiome is a very crucial part of long-term good health for these women. So that's something that we are not focused on right now, but is a good partnering point as a bipartite evaluation of really dealing with PV. At least that's how we think about it right now.
So one thing I want to add, Bethany, is for a phase 1 trial, the FDA is much more forgiving in how things are done. Once you get to a phase 2 trial, which we've now been in, including regulatory agencies in South Africa and the UK, there is a lot more that the FDA is looking for in how stable and consistent your product is. Like any drug, you expect it to be consistent throughout the trial.
Great, thank you. I have some other questions, but I'm going to open it up for. Oh, sorry.
A question?
Laurel,
yes, I have a question for you, Ishita.
Yes.
Because BV is such a multifactorial dysbiosis.
Yeah.
can you produce enough peptides or in your product to cover like multiple different organisms? Because you could, as you said, Adipobium and Gardnerella and Prevotella and staph and maybe other organisms that are there.
Mm-hmm.
How do you
Yeah, so that's where our all of the in vitro efficacy data that we focused on included not just these different bugs, but also a variety of clades of Gardnerella. So, as you may be aware, few clades of Gardnerella are actually now completely resistant to metronidazole. So we are grateful that we were able to obtain a variety of these strains from different resources, including BEI and different strains of this clinical isolates of Atopobium and Prevotella as well, because we want to not just treat Gardnerella because it's all polymicrobial biofilms, and only because we saw efficacy for multiples of these bacteria, even in co-cultures that we were able to make. We want to tend to think that there will be an effect on multiples of these bacteria in the treatment options. And briefly speaking about beyond bacteria also, from the ex vivo vaginal swabs of patients who were symptomatic for BV, but also had comorbidities, including yeast, these peptides did clear out completely Candida albicans, and in some cases, E. faecalis, which is associated often with UTI. So they're more broad spectrum candidates in that sense, which is why we feel that if we have, again, that'll be dependent on a clinical trial, but a 1 or a 4-day regimen may be able to help clear out majority of the bacteria, and then the immune system can take over for complete clearance.
And I had a quick question for Jen about partner treatment.
Yes.
Yeah, it is.
Australia.
Go ahead, Laurel. Get it. Just to mention it, or is there more of a question?
Well,
I interrupted you.
In that study in Australia, many of the men were uncircumcised. Do you think that would hold true for men in the US?
Yeah, that's a great question. So just for folks who may not be familiar with the study, so Kat Bradshaw's group from Australia, the first author is Lenka Bodstrichko, published a study last year of partner treatment for women, so the male partners of women with BV. It was in the New England Journal, and I can try to find the link for folks, which is really exciting. It was very rigorously done and actually showed that it reduced recurrence. There is a great debate currently happening actually in the American Journal of Obstetrics and Gynecology about the context of just some of the specific operational and issues with considering that in other settings, like in the United States, but sort of more broadly about the specific populations enrolled. These were, couples that had been together, so sort of a stable partnership. What does partner treatment look like if it's a, different structure? Circumcision status. We definitely know that there's differences in the penile microbiome of people who are circumcised versus not circumcised, and just a lot of the challenges about thinking about this intervention more broadly in many different settings. So it's a incredibly exciting development in the field, and I think does give us another tool to be thinking about, but is also, I think, really the operational issues of what this means for clinical care in different settings. What does it mean in the context of the US, where we have a very fragmented and even, growingly fragmented system for BV treatment, and versus other settings, and what a partnership structure and some of these biologic issues. So those two, I think, resources it's a really, I think, having the biologic evidence to say like, yes, this works is incredibly helpful. As both of you noted, we've been so limited in treatment options for the past, 5-6 decades, but to have something new is great. But I think that the thinking about how this fits into the larger portfolio is going to be an ongoing and very context specific question.
It certainly makes sense.
Yeah.
Actually, just this last Monday, we talked with an OBGYN who often treats both partners as part of her routine practice, which I don't think is very common, but it's interesting to hear. And just to add to that, for treatment of male partners as well, I did come across couple trials that are done in Brazil and China that talk about men showing up at fertility clinics, having BV markers present, which moves it away from just being a women's health issue and more of a STI besides the NEJM article.
Right.
Yeah.
I think that's a really important, yeah. So we've gotten a couple questions in from people, participants. So I wanted to just see if Tapiwa, great to see you. Do you want to ask your question directly? Otherwise, I can ask it. I don't know. I think it was directed to Ishita. If you wanted to come off mute, otherwise I'll just ask it.
You may ask it directly. Thank you.
Okay, so Ishita, Tapiwa is asking at what pH and temperature range are your products, the AMP, stable?
Okay, yeah, that's a good question also from a translational perspective. We do have really good temperature stability, which is what allowed the formulation for the rapid study of these suppositories at 75 and all the way up to 95 for 10 minutes. Incubation allows these peptides to still retain efficacy. The pH range is also from 2 through 7, where we have not seen any issues in vitro MBC or MIC assays. And that's what was demonstrated even after liquefying the suppositories and extracting active peptides from them. So yeah, 15 months at least that we have tested the suppositories retain fully active peptides.
Great, thank you. And I think, have time for one more question, and then we'll wrap up. And if people want to stay on longer, I'm happy to do so, but I don't want to ask that. So question, I think this is for you, Laurel, from Rhonda Miska. Masika. How often did the product need to be applied during the trial? And maybe that was answered eventually, but maybe you can respond.
We did start with a 5-day loading dose, and then we gave it twice weekly for 10 weeks. Partly because women have sex, and sex can decrease can destabilize the lactobacillus community. And in addition, also menstruation can destabilize the community as well. So we wanted to cover during that time to give women the best chance to colonize. If you do look at the last time point and women then that went on, women who were colonized at the last time point generally continued to be colonized at week 24. Oh, and there was one question about why we used Jensenii instead of Crispatus. And I want to say that Crispatus has an S layer, which makes it much more difficult to genetically modify. We found Jensenii, which is also a hydrogen peroxide producing lactobacilli, to be much easier to genetically modify. There we go.
Well, thank you so much. I want to thank everyone again for participating on this webinar, and the presenters, Laurel, Jen, Ishita. Your talks were so amazing, and bringing your expertise to this conversation.